Hi, welcome to Mathematical Epidemiology. In this video, I introduce you to mathematical modeling. This will be done by Dr. Stephen Mwejande. What to expect to learn in this session? In this video, you will learn what is epidemiology, why we use mathematical models, and basic modeling steps, and the iterative modeling process. Mathematical modeling is the study of disease patterns in a population. There are two main reasons why should we start mathematical modeling. We want to know the distribution of a population or the disease in a population. We want to know who can get the disease, where the disease can spread more easily, when the disease is being distributed in population. We also need to know the determinants of the disease, why the disease happens and how does this disease spread. The main goal is to prevent and control diseases. Why do we really interested in mathematical modeling? Mathematical experiments, most biological experiments cannot be done with real outbreaks. So we need to do mathematical modeling in order to get some insights. Also, we need to predict and plan of disease and how to control the epidemics. We need to compare different interventions. Example, which intervention work more better than the others. We need to understand disease dynamics. The key ideas in mathematical modeling is that all models are simplified representation of reality. The modeling steps. When we do modeling, we follow some specific steps. First step, we identify the problem at hand or the real life or societal problem. We identify the key variable, that's the dependent variable and independent variable. We make simplifying assumptions. Indeed, mathematical modeling usually work on assumptions. So we need to be very clear what assumption we are going to work with when we are dealing with mathematical modeling. Then we write the model equations or the equations for the system which we really formulated. Later, we'll analyze and simulate the model. And here we want to get some mathematical insights about how the disease spread. We do simulation, and here we needed to use some real data or some hypothetical data in order to get some idea of how the disease spread or dies in the population. We interpret the results. Here we write reports, do some recommendations on what we have obtained from the study. As an iterative process, the flowchart here goes like this. We define the problem, we build a model, we analyze the model, we validate the model. Then we try. If we're not satisfied with the validation, we go back to the original problem and restart the process. This repeats until the model is useful. Simple SIR model. Here we consider types. Population. We consider the disease that spread directly or quickly in the population for a short time. Usually we have the susceptible groups. These are compartments. Sometimes we call the infected group, the recovered group. The susceptible, these are individuals who have no disease, but they are likely to get the disease. The infected population, these are the people who are already infected with the disease and they can pass or transmit the disease to others. The recovered people, these are immune. They cannot, therefore, recovered from the infection. They cannot spread the disease to anyone, and they really cannot get the disease because they have acquired some immunity, which can be either a natural immunity or derived immunity from the other sources, for example, vaccinations. The basic equation for SIR model usually is density is equal to minus beta SI, where beta is the transmission coefficients. the output of the S is what the input in the I, minus gamma I. The gamma is just the recovery rate that the individual who is sick, they can recover. They move from S to R, so they're recovering at some rate gamma. The gamma I is the inflow. Those people who recovered, they're joining now the immune people or population. We have the fundamental term or threshold, what we call the R naught. This measures the average number of secondary infections, usually caused by a single, when a single infected individual is introduced in a population which is fully susceptible. For this model, usually it can be given by beta over gamma. Later on, we're going to discuss on how really to derive the parameter. The key points, we have epidemiology. Here is the studies of population, identify the patterns, guides control. The models usually simplify reality, help to predict, and help decision making. Next, we're going to discuss about the types and disease transmission. Thank you very much.